Hey there, I'm Rahina Alfache and I am BS Biology, my major in microbiology, and we are going to discuss about the animal cell. So first up, I have made here, I have made a cell model to better visualize the cells that we are going to discuss, which is the animal cell. And this cell model is a paper Quilled model. So I have gathered sheets, papers that are not used anymore to recycle it and make a cell model, which is this. So this is my cell model. So first, we have there labeled number one, the nucleolus, number two, the nucleus as a whole, number three, the nuclear envelope, number four, the cytoplasm, number five, the mitochondria, number six, the letter 6A. Number 6A, we have here the rough endoplasmic reticulum, 6B, the smooth endoplasmic reticulum, 7, the Golgi apparatus, the pink ones, 8, we have here the vacuole, and 9, we have here the ribosome, very tiny. And number 10, we have here the peroxisome. 11, the lysosome. 12, the centrioles. 13, we have here the green ones, the cytoskeleton. And 14, not last but not the least, we have the plasma membrane or the cell membrane. Oops, don't end this video. We're going to discuss all of them in this video and it's going to be interesting. So, to share with you, I have decided to make use of these papers so that it will be cost friendly and environmental friendly. So, first, let's discuss the cell membrane or the plasma membrane. So, before, okay, so the plasma membrane protects the cell. It is a semi-permeable membrane. It consists mostly of phospholipids, making them a semi-permeable barrier between the cell and its physical environment. It has some protein components in it, and it is very consistent around the cell. All living cells have plasma membrane. So the function of a plasma membrane is of course to enclose the cell content to protect them. They also regulate the molecules that pass into and out of the cell through the plasma membrane. Therefore, it controls homeostasis. The proteins are actively involved in transporting materials across the membrane. The proteins and lipids allow cell communication, and carbohydrates, which decorate both the proteins and lipids and help cells reorganize each other. So, semi-permeable membrane. These blue here, and also consider the white as a semi-permeable membrane, represents the cell membrane. So semi-permeable membrane means that this they do not all they do not allow the molecules to easily or freely go in and out of the cell. Oxygen and carbon dioxide easily goes in and out of the cell, but larger molecules do not. So they need proteins first. So I have here these blue ones here. I have much like imagine that the circle in the middle here is a hydrophilic head, and this the ends of this one molecule here is the hydrophobic tail. So let's proceed. Oh, so this is the cytoplasm. So the cytoplasm, this is a gel-like material that contains all the cell organelles. It is inside the cell membrane. It is enclosed within the cell membrane. So these organelles include mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, and other vesicles and microtubules. So This cytoplasm is it's like this is where these organelles, what I have said late kanina, this is where they float. So this the white hands here, the white ones here represents the cytoplasm. So next, oh, we have the nucleus. So the nucleus are um, they control the control main control of the cell. So first, the nucleus is a spherical structured organelle found majorly at the center of the cell and surrounded by a double layered nuclear membrane, the purple ones here, separating it from the cytoplasm, or separating this nuclear membrane separates the nucleus, the inside of the nucleus, its its environment, or generally the cytoplasm. It is held together to the cytoplasm with the help of filaments and microtubules. It holds other cell organelles, which includes this nucleolus. The nucleus is where the synthesis of ribosomes occur. It also has a nucleosome here, the white ones, or the nucleoplasm. We have here also the chromatin. The chromatin is tightly wounded DNA. It is where our genetic material can be found. We also have here the nuclear pores to regulate the entry and the exit of the molecules in and out the nucleus. So, the primary role of the nucleus is to control and regulate cell activities of growth and maintain cell metabolism. It carries the genes that have heredity information of the cell. This is the chromatin that I have said a while ago, this black string material here, and therefore the chromosomal DNA and genetic materials, which are made up of genetic coded, ultimately make up their proteins amino acid sequences for use by the cell. Therefore, the nucleus is the information center. Now, next up, let us proceed to the mitochondria, the powerhouse of the cell. We all know this. If we question uh, other students, they all think of mitochondria as the powerhouse of the cell because it is true. It is a rod shape. As you can see, it is oval shape. Mitochondria have two special membranes. Here, the outer membrane. Oops, parang naano ata to. Ay, sorry. Never mind this chromatin here. I guess it was ano. We have here the outer membrane and the inner membrane. So the membranes bend into fold known as cristae here. As you can see, they have bends here. Ayan. So their primary function is to generate cell or energy for the cell. They are the power generators producing energy in form of adenosine triphosphate or ATP by converting nutrients and oxygen into energy, enabling the cell to perform its function and to also release excess energy from the cell. Okay, as you can see here, we have the mitochondria ribosomes. It has its own DNA. Yes, it has its own DNA. And the ribosomes and the DNA are located at the matrix, in the matrix, from the matrix of the mitochondria. You can also found in between the outer membrane and the inner membrane, the intermembrane space. Now let's proceed to the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, literally, this is our continuous folded membranous organelle found in the cytoplasm made up of a network of flattened, as you can see here, flattened interconnected compartments or sacs that connects from the cytoplasm to the cell nucleus. So, the smooth endoplasmic reticulum is not associated with the ribosomes, unlike the rough endoplasmic reticulum. We will show it later. which is covered with ribosomes. And their action is different from that of the rough endoplasmic reticulum. Despite lying adjacent to the rough endoplasmic reticulum, they function differently from it. It is responsible for the synthesis of lipids and that are utilized for producing new cellular membranes. They are also involved in synthesis of steroid hormones from cholesterol for certain cell types. Now let us proceed to the rough endoplasmic reticulum. As you can see, they differ from, although they look closely the same, these rough endoplasmic reticulum have ribosomes on it, attached on it. So we have here the cisternae, the cisternal space, we have the ribosomes, and we have here a transport vessel because rough endoplasmic reticulum functions for the synthesis of proteins. I mean, the ribosomes in the rough reticulum functions for responsible for the synthesis of proteins. If nucleolus is responsible for the synthesis of ribosomes, ribosomes are responsible for the synthesis of proteins. So rough endoplasmic reticulum transports the proteins and lipids through the cell into the cristae. So they are sent into the Golgi bodies or inserted into the cell membrane. So speaking of Golgi bodies, these is a Golgi body, or the Golgi apparatus or the Golgi complex. These are membrane-bound cell organelles found in the cytoplasm of a eukaryotic cell, next to the endoplasmic reticulum and near the nucleus. Golgi bodies are supported together by cytoplasmic microtubules and held by a protein matrix. It is made up of flattened stacked pouches known as cisternae, like in endoplasmic reticulum and endoplasmic reticulum. So they have three primary compartments known as the cis, the medial, and the trans. So as you can see here, this is the cis, this is the medial, and this is the trans. So the cis is the nearest to the endoplasmic reticulum, and the trans is the farthest from the endoplasmic reticulum. And the medial is the central layer of the cisternae. So as you can see here, let us go back to the first picture. As you can see here, this is the cis phase, which is nearer to the endoplasmic reticulum, unlike the trans part. Okay. So the primary function of the Golgi apparatus is to transport, modify, and pack proteins and lipids into the Golgi vesicles to deliver them to the target sites. Animal cells contain one or more Golgi apparatus, while plants have a few hundred. So as you can see here, this is the incoming transport vesicle, and this is the secretory vesicle, or the palabas. Ayan. So let's proceed. We have here the vacuoles. Vacuoles can or cannot be um, found in the animal cell. It is rarely found in an animal cell, unlike the plant cell, which really has a vacuole and and definitely larger than the vacuoles of the animal cells. So they are membrane-bound sacs found also within the cell cytoplasm. The vacuole sac has a cell membrane surrounding, as you can see here, the darker shade of blue, single membrane surrounding it as a tonoplast, and this membrane resembles the plasma membrane. So this is the tonoplast. Their primary function is to store food, water, carbohydrates in the form of sugar, and waste material. Next, we have the ribosomes. The protein, responsible for manufacturing, for synthesizing of proteins. So ribosomes are made up of ribosomal proteins and ribosomal RNA. In a eukaryotic cell, ribosomes constitute half ribosomal RNA and half ribosomal proteins. Yeah. So they are used especially in the central dogma transcription and translation. Now let's proceed to the peroxisome. So peroxisome can also, like vacuole, can or cannot be found in an animal cell. They are spherically shaped, as you can see in the model, bound by a membrane, and they are the most microbodies in cell cytoplasm. The peroxisomes, this crystalline core inside the peroxisome is not always present. The peroxisome's function include lipid metabolism and chemical detoxification by moving hydrogen atoms from various oxygen molecules to produce hydrogen peroxide, hence neutralizing body poisons such as alcohol. Next, we have here the lysosome. The lysosome is like the garbage collector inside the cell. This is a site for digestion of cell nutrients, excretion, and cell renewal. Lysosomes break down macromolecule components from the outside of the cell into simpler elements that are transported in the cytoplasm via proton pump to build a new cell materials. I always remember lysosome as suicidal bugs, but suicidal bugs is the term suicidal bugs is like it's so sensitive for me. Now let's proceed to the centrioles. This is a um this is the most favorite part of the cell model that I have made. So this it the centrioles is a small structure, and it is made up of 9 sets of microtubules. As you can see, let's count it: 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 triplets, 9 microtubule triplet. As you can see, they're they remain very strong as triplets, and hence they have been observed to be in structures like cilia and flagella. The triplet microtubules are held together by proteins, giving the centriole its shape. The centriole microtubules allow the transportation of substances that are linked together with a glycoprotein to any cell location. The glycoprotein linkage acts as a signaling unit to move specific proteins. These centrioles um, is has a very important part during mitosis. So let's proceed to the cytoskeleton. Ayan, so the cytoskeleton provides support for the cell. So okay. It functions as a network organizing the cell components and to also maintain the cell shape. So para hindi makalat yung mga organelles. So they are the ones organizing it. Kasi palutang-lutang lang yung organelles sa cytoplasm eh. It also provide a uniform movement of the cell and its organelles by the filament system network found in the cell cytoplasm. It plays a major role in the movement of the cell and some organelles in the cytoplasm. And that's it. That's the end of our discussion for the animal cell. I hope you learned something, and I learned something too. Not just something, I learned a lot. Thank you very much for watching. I hope you proceed to the next video. which will be the plant cell discussed by my groupmate Ryan Carlo Fajardo, and I hope after watching his video you watch our discussion with me, Ryan, and Renz. Thank you so much and have a nice day. Thanks for watching.